Good morning. Thanks for everyone for sticking through this. As everyone else has said, it really is a great honor, particularly as I'm not quite as embedded in Li-Fraumeni syndrome as many of the other speakers, so I'm really not going to try to duplicate a lot of what we've learned this morning. And I took the in children off because I decided that for this group it would actually be really useful to talk about some of the efforts going on in ClinGen and Clinvar and how we might also engage the association itself. So, and just there's been jokes about the Sharons, but just to confuse things further, I'm going to talk about Sharon Savage's work. So first, to just set the scene, many of my scientific colleagues know that I was involved in a study that was just completed where over 4 years we offered exomes, so very comprehensive genetic testing to almost 300 newly diagnosed childhood cancer patients, all solid tumors and brain tumors, independent of any family history. And we did identify that approximately 10% of those children carried an important germline susceptibility mutation that might have implications for them or their families. So with regard to Christian's comment, we are now doing a bigger study in Texas, and certainly it's my prejudice that as a medical geneticist that relatively soon we're going to start offering genetic testing to all childhood cancer patients. But one of these patients kind of highlighted the problem that led to me to be involved with the clinical genome resource, which is we had one of the very early children in the study, a 5-year-old boy who had been diagnosed with adrenal cortical carcinoma, was enrolled in our study. If you just looked in the medical record, there was really no significant family history. But the whole exome sequencing, and again, this was back in 2012, revealed a missense variant in TP53. The clinical laboratory at the time reported it as a variant of uncertain significance, but I told the family that given the clinical scenario, the adrenal cortical carcinoma, it was likely the cause, but that the current clinical practice, has to do with the fact that the laboratories at that time would only report it as pathogenic if it was already in the database that way. Actually, after that identification, it was subsequently identified across actually several states that this family actually has multiple members with cancer who carry the same variant. And it is now submitted once as likely pathogenic by another laboratory in Clinvar, as well as our submission. So there is this huge issue with variant interpretation, and that's what's really the topic of my talk today. So first, I just want to talk about Clinvar. Clinvar is a database, so this is for the U.S. people here, your U.S. taxpayer dollars at work. This is a database that's hosted by one of the National Institutes of Health, in this case NCBI, the Biotechnology Institute. And there's a very nice publication describing it. Melissa Landrum is still a major player and the chief staff officer for it. And these numbers are actually old, but there were already 512,000 submissions, over 300,000 unique DNA variants across clinical genes. And I just want to point out it's a very international database. There are 824 unique submitters representing 60 countries. And I looked up, and it's all publicly available, so while sitting here yesterday writing my talk, I looked up P53 in Clinvar, and you can see that overall, there are 1,200 unique variants in P53 in Clinvar, and you can see that 143 of them have conflicting opinions from the submitters, but you can see the various breakdowns of them. So this, we think, is an important resource, and we certainly recommend anyone representing laboratories here submit your data. And I'll talk at the very end about how people who may have had testing years ago can submit their own data as well. So what's the issue? And this is just the variant that I was talking about, to just say that if you look up any given variant, this is just some of the information that's provided. Ambry Genetics, I believe, is the other laboratory that submitted it. And we submitted all the data from our study, and that's why it's there as well. Okay, so what I want to spend the time on is really talking about ClinGen and how it works with Clinvar to get really much more as an authoritative central resource to really try to define the clinical relevance of genes and variants It is primarily funded by the NIH. There are 3 major grants, and I'm co-principal investigator of one of those grants with Carlos Bustamante at Stanford. It started in 2013, and I'm happy to say it was renewed for 4 more years of funding. So we're now in year 5 of a total of 8. And there are really well over 500 people on the different committees. And again, it's highly international. And pretty much everything you want to know about ClinGen is at clinicalgenome.org. And this is just our website. So what do we really do? And I'm really going to focus on the second question. We have 3 main questions. One is, what's the data that a gene's associated with disease? And there's plenty of data that P53 is associated with disease. Then what the bulk of the work is what's the evidence that this variant that we're looking at actually causes that disease. And then there's another whole group in reference to the last talk, really, or the talk before it, really looking at the question of what's actionable. So which of these variants should we really take medical actions? But I'm going to focus on P53 variants and what we're trying to do with regard to that. So what we set up are what are called clinical domain working groups, and I'm co-chair of the hereditary cancer one. We have a whole bunch of experts, and then each group spells out these different committees that really try to do expert curation on important genes. And we also have help from professional societies, clinical research organizations, other academic institutions, I should say representatives from many diagnostic testing labs, both commercial and non, are members of our committees. And we really try to bring in the whole kind of genetic testing, genomics environment. So one of the important things to know is when you look up a variant in Clinvar, that working with ClinGen, we've tried to create some guidance as to how good is the evidence around that variant and what we say about it. So the top would be a practice guideline. There are actually very few of those. For example, there's a guideline about exactly which cystic fibrosis variants to test for. But the bulk of what we're focused on is this expert panel group, trying to get experts for a given gene to get together and really use a systematic approach to decide which variants clearly are associated disease, that's called pathogenic, which are clearly benign, and which remain uncertain. 1 star is also very important. These are the submitters, often clinical laboratories, that really tell us how did they come to the conclusion they came to on that variant. And then there's certainly a variety of others. Okay, so what are the key features of the expert panel? It has to be multi-institutional in membership, often international. We need to have both clinicians who take care of the patients, laboratories that report the results, and scientists. I have to say, so far we haven't had advocates, but certainly that could be a possibility. Generally, you really want to focus on one area. What are you guys really expert on? We've had people apply for like the whole exome. We're like, no, you're not an expert on all 20,000 genes. And you have to be public about how you're doing your variant classification procedure. You have to be willing to be reviewed. And in fact, we had some early expert panels that already existed. For example, Enigma, which a number of you are involved with, which deposited for BRCA1 and 2. Okay, so what's key here is that the hereditary cancer group kind of looked at the environment and said, like, what are the important genes for which there's not already a group? And obviously P53 was one of the very first. And so both P10, CDH1, and P53 were the early groups that our committee put together. I do just want to point out that myeloid leukemia is a brand new one, which the American Association of Hematology, American Society of Hematology is supporting in partnership with ClinGen. And so this is an example of us partnering with an outside organization. Similarly, Raymond Kim, or at least he was here yesterday, is leading the VHL effort, and he's worked with the VHL Family Alliance to get some support of some of his work curating those variants. So I actually think I'm going to skip this for the moment because of time. We do use the American College of Medical Genetics and Genomic Variant Classification. This just shows all the different kinds of data that you have to look at for each variant to come to a conclusion. We provide some guidance about that. We provide some guidance about which ones not to use. But then really the expert panel looks at all of these squares, from particularly the ones in purple, from the perspective of how is that relevant to my gene. So, and these are the steps to get approved. So I'm happy to say that Sharon Savage now, I think probably almost 2 years ago, or a year and a half ago, agreed to chair the P53 variant curation group. Christy Lee, who's a genetic counselor working through the UNC ClinGen grant, has coordinated that and played really a critical role. There are 22 members from academic, governmental, and commercial institutions from really around the world. I'll show the full list on the next slide. And they were divided up into kind of 3 subcommittees to tackle those different squares on the prior slide. So the different parts of the ACMG criteria are looked at by these subcommittees, and then they make recommendations to the larger committee to come to a set of defined criteria that you'll then look at the variants and come to a conclusion. This is, in fact, the longer list of people on the committee, and you can see a number of them are here today. So what have they been working on? And really, just to reiterate again, the goal of this committee is to use our current knowledge of P53 and Li-Fraumeni syndrome to really specify and improve the rules for variant classification, and then make those classifications public through the ClinGen webpage, as well as through submitting them back to Clinvar. So there's kind of a cycle here. Variants get submitted to Clinvar by diagnostic labs and research groups. That data is then used by the committee to classify the variants, and then those submissions will go back, once they're approved as an expert panel, will go back into Clinvar so that it's available for the community. And so why do you need a committee? So part of the problem is that if you look at the ACMG rules, you need to be able to say, what do you really consider a Li-Fraumeni phenotype? What do you really think is the incidence of P53 variants in the general population? So a lot of the data we've been hearing at this meeting is data that the committee's using to come to conclusions about how to classify variants. So this, for example, is just the conclusion they came to for using a strong evidence code versus a moderate evidence code. This is kind of ACMG speak about how do you apply the rules. And so this is just an example of the kind of step-by-step work. So this is not necessarily fun or very exciting research work, so I'm very gratified the committee's willing to do it, but it's actually really important work. So for each of these codes, you have to decide as a committee, what do we think is a convincing phenotype? So you can just see here that they define exactly which kind of tumors, based on the prior slide, would get you this strong evidence code versus this moderate evidence code and things like that. And then those codes are all combined, and you get a final readout of pathogenic, likely pathogenic, uncertain, et cetera. And this, again, is similar. For the phenotype, there's a code that says, okay, so take my patient. I had a patient with adrenal cortical carcinoma who had that variant. Would you meet this criteria or not? And so they decided that you had to actually meet Champray criteria for them to apply the code. So the committees are often quite conservative because they don't want to overcall variants in doing this work. One of the things that ClinGen's provided is a bunch of tools to try to make this easier to do. The team at Stanford and at Baylor, the informatics teams, have built these web-based curation interfaces. They're all open to the community, so you need logins, but you can request logins. So if any of you work for laboratories that want to use any of these tools, they are available for your own group for curating variants. It walks you through the different pathogenicity codes of the ACMG guidelines. Okay, so just to say kind of where they're at, and I apologize, Sharon may comment better, I think this is actually slightly out of date, but generally what these groups do is they pick some gold star variants, so variants they know are pathogenic, variants they know are benign. They use those to test the rules. Then to test out the rules, they have 2 people independently curate the variants using the rules that they're trying to specify. They look at how they do, and they modify them as needed to come up with a set of codes, which will then be approved by the ClinGen Steering Committee. And they're doing this in the variant curation interface that I mentioned on the prior slide. So they're kind of in the midst of this. They will then submit those rules for kind of final approval before they actually become an expert panel and then start curating larger number of variants and submitting to Clinvar. So the last thing I wanted to just talk about, especially for the families, is there's something else that ClinGen has created, which is called Genome Connect. Now, there are a number of patient registries, and I know some of the people here have worked on PROMPT, and there are a couple of others. This is actually not disease-specific. Genome Connect is open really to anyone who's had genetic testing. And again, you just go to genome.org, and you click on the Genome Connect button, and you get this very pretty page, and it tells you all kinds of questions and frequently asked questions, but essentially it allows you to enroll remotely, and if you have a copy of your genetic test report, you can upload it. It gets very carefully looked at by a team of experts. Your name, of course, and all your identifiers are removed, but then that data can be submitted to Clinvar. So for example, if you had testing 10 years ago, before Clinvar even existed, that laboratory might not submit to Clinvar. It does allow you, they also actually ask you health questions to answer, and there's a whole informed consent, so they talk about privacy and all those kinds of things. But it does allow the public to provide data. They do also, on the next slide, I'll show you how they also partner with other organizations. And so they also, I think, would be happy to partner with LFSA or any of the Li-Fraumeni if you wanted to set up a registry to allow this. So this is just an example of a poster they gave at the American College of Medical Genetics last month, I guess, when they looked at some of the initial submissions from people. So again, this is not P53, this is all different kinds of genetic diseases. It turns out that about half of the genetic test records that were uploaded, in fact, that variant does not appear in Clinvar. So no other test laboratory had yet submitted it. So this is new information for the field that we might not otherwise had gotten information. For the other half, the patients telling us about their own disease actually now provides important phenotype information. Even if the variant's already in Clinvar, those individuals providing that information really give us even more information. And this just kind of tells you where these came from. The right-hand side of the poster, which again, you can download the poster, we're very good about making things as public as possible, is that they're really interested in partnering with other organizations. And so this is just, if you already have a registry on the same platform as Genome Connect, it's even easier. If you don't, there are ways that you can look at data sharing, or they could create some kind of registry with you. I mean, I'm not making any promises here, but they're definitely looking for partnering with other organizations, as well as like our partnership with ASH, getting partnership with organizations that want to support curation of the genes they care about. So moving forward, I just have to say personally, I'm incredibly gratified by the efforts of the community to join and work on the ClinGen P53 expert panel. I just want to be clear that the vast majority of the people on that panel are not being paid through any of the ClinGen grants. They're really doing it from their own interests. This is my last slide, should be okay on time. We're sharing of clinical test data via Clinvar or other databases. I apologize, I forgot to mention that the IARC P53 database is represented on the panel and has been really instrumental in providing data for the curation efforts. The sources of funding over this time from cancer-based organizations is critical. NCI is not currently funding ClinGen, and any of you who have any buddies at NCI, we would love to see that, to sustain the effort and accelerate variant classification. And as I mentioned, Genome Connect or other patient portals would really benefit from some of the information that the families here have who've already undergone testing and are known to have Li-Fraumeni syndrome with an E. Thank you. And I just want to acknowledge all of my colleagues in the ClinGen effort.
Thank you, Sharon. We can take one question, and then we'll have to move on. Please. Jake.
I'm a genetic counselor from Japan. I just would like to know, And once it's classified as a variant of uncertain significance, and then of TP53, then what's going to happen? Because I remember that in terms of BRCA1 and 2, Myriad used a lot of commercial budget to have segregation analysis to reduce the VS percentages. So it was
so I think the question is about what do we do about VUSs. So actually one of the important effects of the ClinGen committees is that variants that really have only been seen once by one lab are likely to just stay a VUS. But often if you look in Clinvar, you can see a variant that's been listed as a VUS by 4 different labs. And we've already seen this now with like the CDH1 committee, that if those 4 labs now combine data, you can actually now say, oh, this is actually likely benign. Or actually, if you look at those 4 together, those 4 families together, you can actually shift to pathogenic, or likely pathogenic. So I think that's one of the advantages of the data sharing. The truly unique variants, first time ever seen, it's extremely hard to say anything but variant of uncertain significance. But often you have 5 different labs that each say, I've only seen it once. Together, that often is enough data to then classify the variant. Thanks.